Everyone. It's my pleasure to introduce our first speaker, our first keynote speaker of this morning session, Dr. Anthony Guiseppi-Elie from Anderson University, where he is a distinguished university professor, the vice president for industry relations, and the founding dean of the College of Engineering. At Anderson University, Dr. Guiseppi-Elie currently directs a Center for Bioelectronics, Biosensors, and Biochips. And before that, he was the department head of biomedical engineering and the founding associate dean for engineering innovation and engineering medicine at Texas A&M University. He is also a founder, president, and scientific director of the biomedical diagnostics company Abtec Scientific Incorporation. Dr. Guiseppi-Elie started his education at the University of West Indies. He then moved on to receive a master's degree in science in chemical engineering from the University of Manchester Institute of Science and Technology. And he received his doctorate in material science and engineering from MIT. Dr. Guiseppi-Elie holds 8 patents and is a fellow of the Royal Society of Chemistry and a fellow of the Institute of Electrical and Electronics Engineers in recognition of his contributions to the field of bioelectronics in particular. So Tony, we're so glad to have you present and be our first keynote speaker. We very much look forward to your talk today. Thank you.
Thank you so very much, Chris. It's indeed an honor, a great pleasure, and an overwhelming delight to be able to meet with and present to this group. I thank you very much for the invitation, Dr. Wong, and to the organizing committee for the invitation. And of course, to our organizing group, Hoover, and to Miranda Cole, who has done a phenomenal job in pulling all of us together. I want to talk a little bit about work that was ongoing, that began at Clemson University while I was a professor at Clemson, moved to Texas A&M, and now it's being expanded in some broader collaborations, which I'll talk about. But it's really around the notion of fusing biomarker data to come up with quantifiable scores that reflect the status of a patient. And one of the challenges we face is in hemorrhagic trauma, that is trauma that leads to bleeding. We'll talk about this at length. Again, my great honor and pleasure to be part of this conference and to present this work. Okay, I'm frozen. So one of the things I have to do is disclose to you that I work for a company as well. I have founded this company back in 1995. That's the holder of some of the patents that we produced, and that this company continues to operate in the space where it uses bioelectronics type devices to do diagnostics. So today I want to talk about hemorrhagic trauma, and I want to talk about a biosensor system we call the PSM biochip that really attempts to collect physiologic status data from a hemorrhaging patient, and then how we might use data analytics and predictive analytics to sort of stratify those patients for intervention. As part of that work, we had to come up with a digital patient. As part of that work, we had to come up with a score to which physicians could react. And we had to come up with EI approaches to enable that. So we'll talk about these things. But first and foremost, of course, this is work that has been ongoing for a while and has been wonderfully supported by students over the years. Dr. Kutanen did his PhD with me at Clemson. He's now a resident at Walter Reed National Military Medical Center. And Dr. Karunway did his PhD also at Clemson. He's now an assistant professor at Anderson University. My most recent PhD, Dr. Bat, completed her work at Texas A&M and is now an Intel postdoctoral fellow. I'm also talking a little bit about work of another recent PhD, Dr. John Agus, who is now at Roche Diagnostics. So my hat's off to all these wonderful, talented young people who had an opportunity to come and work with me. It's their work that shines, as we'll talk about. So hemorrhage is one of the leading causes of death amongst individuals who are less than 46 years of age. Thankfully, that's not me. People above 46, of course, die from other things. But when we talk about hemorrhaging trauma, 62% of all deaths are in those age group 15 to 24. If you're a young person, you're likely to die from trauma, a fall, an accident, gunshot wound, or etc. And it's the highest contribution to death rates within the first 24 hours. In other words, within the first 24 hours, most patients will suffer some demise, will come to demise. In general, we talk about the golden hour, which by our observation is typically about 45 minutes when major hemorrhage ensues. A fall that hits the back of the head that leads to bleeding in the brain can take a patient to sleep and not wake up. The recent case of Mr. Saget notwithstanding. But what happens when we bleed? When we bleed, one of the consequences is a very complex sequence of events that occur at the molecular level, through the tissue level, through the organ level. The principal amongst these is vasoconstriction. That is, the peripheral blood vessels constrict, limiting blood flow to the periphery and keeping blood in the core. The consequence is there's a very high oxygen demand in the musculature. And it turns out that the musculature is one of the largest contributors to signaling molecules, because they are the ones that most profoundly are impacted by vasoconstriction. And most of the oxygen that you consume is consumed in the musculature. So your muscles are the big working juggernauts in the human body. And when we bleed, we limit access to the muscles, preserving blood to the core and to the brain. But what we learn about this is, of course, is that it can lead to cell hypoxia. That is, indications that cells become hypoxic. They undergo a kind of anaerobic oxidation. We see the emergence of lactic acid as a key indicator of the status of this process of consequence of vasoconstriction and anaerobic oxidation. And we know, of course, that lactic acid is linked to glucose metabolism via the Cori cycle. And that Cori cycle, noteworthy, first described by Carl and Gertie Cori back in 1928, I think. They won the Nobel Prize in 1947 for this work. This is a linked cycle between glucose activity and lactate. And so what we do know is that one goes up and one goes down. And we see these as opportunities for measurement and monitoring to generate data. The second area, of course, is that the lactic acid results in metabolic acidosis. That is, there's a pH increase in the musculature. And with lots of rationalizing, we see that cells that become hypoxic eventually release potassium into the extracellular space, which then makes its way into the vasculature. So we see this spike in potassium. These are examples of analytes, metabolites, or biomarkers, as we call them, that may be indications of the status of a patient who has begun to bleed and may ultimately suffer what we call multiple organ dysfunction syndrome, or MODS. Multiple organ dysfunction syndrome simply is an indication that the patient has moved towards hemorrhagic shock, and all of the organs of the body, the kidneys, the liver, the spleen, etc., begin to experience some dysfunction. Well, we've talked about analytes, and the reason we've identified these analytes is because we see them as a rich source of data. On the left, you see glucose, lactate, potassium, pH. We talk about our motivations for why we may want to monitor these. We talk the methods by which we monitor these and the techniques that we're going to use. to monitor these. Remember, our purpose is to collect data that informs the status of the patient so that we could now use that for stratification. That's the context. The technique we will use for both glucose and lactate is a technique called amperometry. It's an electrochemical technique wherein we apply a voltage, we measure current, and we can do this under different conditions. The transducers that we will use are called biotransducers because they involve the use of an oxidoreductase enzyme on a microdisc electrode array. So those are going to be microlithographically fabricated. We're going to look at potassium. Potassium, we're going to monitor using potentiometry, which is measure of voltage versus some sort of reference. We're going to look at pH using a pH-responsive hydrogel, and we're going to measure that by impedimetry. That is, we're going to measure a transfer function after having applied a sine wave. And we're going to use interdigitated electrodes for that. Now, oxygen tension is a little bit more challenging to do. We're going to do that with voltammetry. Of course, you know, you can use a pulse ox. It uses an optical technique. But when you're indwelling, we are going to use a voltammetric technique. We're going to sweep a voltage, extract a current, and we're going to use a microdisc electrode for that. The key thing here is that we wanted to have a set of compatible measurement modalities that we could identify in the form of a microfabricated device. This is what that device looks like. It's big on the scale of things in electronics. It's 2 millimeters by 4 millimeters, but it's intended for research purposes and not for commercialization. But here I point out some of the key features of this device. The two areas of interest are, of course, the glucose sensing and the lactate sensing, which uses a microdisc electrode array. What you're looking at is an array of micro discs wherein a metallized substrate is patterned, and that metal could be gold or platinum iridium, and then it's passivated with silicon nitride. We then etch through the silicon nitride to reveal the metal beneath. And it's at that metal that we will do some chemistry and biochemistry to make these measurements. The interdigitated array is similar in that it's also microfabricated. So the process of microlithographic fabrication generates all the types of devices that we need on one substrate. Interdigitated, the critical feature size here is 1 micron. These lines are combed, meaning the electrodes are like combs that are interfingered or interdigitated. And the separation distance between these lines is 1 micron. We have a potassium sensor, which is a microdisc electrode. It's 10 microns in diameter. The oxygen sensor, similarly, is a 10 micron disc with some very interesting decorative chemistries. In all of these cases, we are building biosensors and biomedical sensors. And the distinction is that in a biosensor, such as in glucose or lactate, we're using a biological molecule to confer recognition and sensitivity. In a biomedical sensor, we may be using a material, a responsive material or a responsive surface to measure something of biomedical interest, pH, potassium, oxygen. But there is no molecular recognition that involves a biological molecule involved. So the 5 key analytes of hemorrhagic trauma are being measured on this device. We can build potentiostats, potentiometers, and impedance analyzers, and on scales that are compatible with animal studies. So we can multiplex these, of course, simply switching and tapping between. And the key is that multiplexing in this case works because the update frequency for each of these sensors is on the time scale of minutes over a 45-minute period of indwelling. So the goal is not in microseconds or even milliseconds, but to update on the order of minutes. So multiplexing is a useful approach here. But let me say a word about this glucose and lactate biosensor, because I think I want to give you a sense that this it's a non-trivial task. In the technique I described in amperometry, we simply apply a voltage and we measure current, and that's been a technique that's been around since the 1800s. But increasingly, as we apply these to biological phenomena, we have to engineer the interface between the device and the biology. And one of the features that we do here is to use these microdisc electrode arrays, which enhances the current from the diffusion flux, because we now have discs that are on the order of 25 micron critical feature size. So we engender radial diffusion, we increase the flux of the species of interest. You see that C star, the concentration. And in this case, it's glucose or lactate. So here are the simple biochemical reactions. And I know at 9 a.m. in the morning, no one wants to really study biochemistry, but glucose oxidase is a very simple oxidoreductase enzyme that has the capacity for recognizing and transmuting glucose to gluconolactone, and it generates hydrogen peroxide. So in the simple type generation 1 biosensor, the peroxide is reduced, we get a current. So that's simple, and that was described back in the 60s of the last century. Point is, hydrogen peroxide is a powerful oxidant. It is capable of oxidizing the very enzyme that produced it. And in nature, these oxidoreductase enzymes are usually in very close proximity to peroxidases that remove the peroxide very rapidly, converting it to water, and eliminates the formation of sort of reactive oxygen species. So, but the key is generation 1 type devices may be used. We don't find them particularly useful. The generation 2 type devices that I show here in the middle is the type of device that is mediated. That is, we use some sort of intervening chemistry where we poise the potential of the redox reaction you're associated with the enzyme to some other species, such that the electron cascade is thermodynamically favorable, and it results in a mediated electron transfer. In this particular case, we're using a redox polymer. And that redox polymer is an inherently conductive polymer called polypyrrole. There are a large number of other polymers that may be used in this group, but the polymers we have worked with are copolymers of polypyrrole. The nice thing about this is that you can do it both amperometrically or impedimetrically. The third is a third generation type sensor in which the electron transfer reaction results in a direct electron transfer between the FAD of the oxidoreductase enzyme and let's say a carbon nanotube or a carbon dot or multi-ball or single ball or graphene. And we can build these direct electron transfer type devices that eliminates the need for the production of hydrogen peroxide in the first place. So these are highly favored in indwelling devices. Well, we can build these sorts of things, build the instrumentation, work with commercial sources to identify potentiostats, impedimetric devices, and indwell these in animals. So the chip that we've just described is internalized, and the hardware is externalized in our animal studies. We could design hardware around the potentiostat, the potentiometer, the impedance analyzer, where we have amplifying circuits. We use Bluetooth. Power is a major consideration, so the management of power on these devices to allow for these things to last for about at least 5 days is critical. So all of this hardware is externalized. The battery life is around 5 days. We have a footprint of about 2 times 5 centimeters, which is pretty big, but that sits outside of the animal. And we are employing some intelligent power management. So that's what that device looks like. Now, so the instrumentation, it turns out, is the easy part. The hard part is building the biotransducer and designing that biotransducer so that it's stable, non-drifting, doesn't biofoul, gives you the sensitivity that you need. And by sensitivity, of course, I'm referring to the dose response curve that you see on your screen. We're looking at the steady state current versus the concentration of an analyte, and that gives us what we call the dose response. You see in black here two lactate biosensors, one with a very high sensitivity, high slope, with a very low dynamic range, say from about 0.1 to about 5 millimolar. The other has high sensitivity, a low sensitivity, sorry, but a wide dynamic range, going up to about 17 millimolar. Well, the challenge for us in biosensor development, of course, is to engineer the mix of transport with mediation and response to get the sensitivity to be just right. In the case of glucose, that is a good example of a well-designed sensor shown in red. So I mentioned that we could do the generation 2 type biosensor with impedimetry, and here I show an interdigitated array reacts with a conductive polymer film that's oxidized by the hydrogen peroxide that's produced, and that leads to a change in conductivity. That change in conductivity can be monitored and has to be monitored usually with a very discrete sine wave, small amplitude that extracts the transfer function to yield the impedance. I share this with you simply to let you know that this is work that we've been doing for a while. Here is an example of an early patent back in 1994 that described sort of this general impedimetric concept for mediated glucose response through an impedance change in a conductive polymer. And so one of the key points I wanted to make is, of course, that the goal is to impact patient care. And the path forward from any of our research is through creative invention and eventual commercialization to bring good things to the clinic. And so that the path leads through patents. I want to point out that the generation 3 type biosensor that we developed was developed in a most uncharacteristic way. We knew that we could incubate carbon nanotubes with enzymes, which are surfactants in nature. That is, they have a water-loving outside and a hydrophobic somewhat core. And we could incubate those with carbon nanotubes, and the hydrophobic carbon nanotubes, single-wall carbon nanotubes, or graphene sheets, the proteins would adsorb. It'll take a long time, but eventually they would adsorb. We found that we could accelerate this process by using ultrasonication. So we undertook a very comprehensive study of how to look at the use of tip sonication to accelerate the process of integration of the carbon nanotubes with the enzymes. We could then do ultracentrifugation, extract the appropriate layer, which had the conjugates of proteins with nanotubes. And we could then use those in some very interesting studies. So we were able to show that, for example, a carbon nanotube GOX conjugate would show an electrochemical profile in phosphate buffered saline that looks like the profile shown in the gray box. That is, the cyclic voltammogram of current versus voltage would look like this, and that the enzyme, the cofactor itself looks similar. That was evidence that we had successfully connected the nanotubes with the cofactor. And more importantly, under oxygen poor conditions, anaerobic conditions, we were able to dose respond the enzyme conjugate. We went on to do lots of characterization of those systems and published on that extensively. But here I show now an impedimetric response to a glucose oxidase carbon nanotube system in a polypyrrole matrix, the polypyrrole hydrogel. But the key thing I want to point out is that look at the control experiment where we're extracting the impedance magnitude Z versus time following a series of oxidation, reduction of the conductive polymer. And each of these in the sawtooth pattern is us reducing the polymer and letting the polymer reoxidize in the presence of the peroxide that's generated. So what you see here is the system based on generation 1. And what you see in green is a system based on generation 3. And in red is a hybrid of the two. We were able to show that we can do a hybrid Gen 3, Gen 2, and get the most significant response as shown in the panels beneath. So the red curve shows you the dose response curve for a change in impedance versus concentration that has the highest sensitivity. And we can do, of course, all of the characterizations of these types of systems to describe the enzyme characteristics under those conditions. So we set about doing animal studies. So I belabored a lot of chemistry and biochemistry simply to tell you that we did a lot of work characterizing these systems before we began to put them into animals. So here you see an animal that has an externalized bit of instrumentation on the head and the internalized sensor in the body. So one of the things we do to simulate hemorrhage is simply to draw the animal's blood out. And I apologize to anyone with sensitivity. I should have said that you will be seeing a couple animal studies experiments. So if you have some sensitivity to the observation of surgical procedures, I advise you to turn away. Here is a sensor that's internalized, and here we've put a catheter that allows us to draw blood out of the animal and reduce its mean arterial pressure. The mean arterial pressure is shown in green, and we're drawing blood down so that the animal is is hemorrhaging. We're looking at, in blue, the amperometric current to the tissue lactate, that is the lactate that's in the musculature, and we're looking at the lactate that's in the vasculature, the so-called blood lactate. Well, immediately you can see that intramuscularly, the lactate levels are rising faster than in the vasculature. Just as I indicated, the lactate level, anaerobic oxidation in the musculature leads to lactic acidosis in the tissue, in the muscle, before it appears in the circulatory system. Of course, most physicians draw blood and get stat response on lactate. So the point here is that what you see in the vasculature may in fact be misleading with regard to what's in the muscle, where the site of lactate generation. We did a lot of animal studies confirming this, and I shan't bore you with this. And we did lots of animal studies where we spike the animals with the source of glucose, for example, by feeding them Ensure. I drink Ensure, I just let you know. And we monitor what goes on intramuscularly versus intravascularly. Our potassium sensor was really quite interesting based on old school approaches using valinomycin, which is a circular peptide that has a high binding efficiency for potassium. The way we did this, of course, is somewhat unique. We can develop a potentiometric response by immobilizing the valinomycin into a conductive polymer film that was electrodeposited onto the electrode. It's a little bit more chemistry than you here to cover at this time. We were also able to build voltammetric sensors for oxygen by simply using underpotentially deposited platinum onto the platinum electrode. And by underdepositing platinum, we create platinum nanoparticles that A, were far more catalytic to oxygen reduction and far more surface area, so they enhance the current response. Again, because it's a 10 micron disk, we're supporting radial diffusion of oxygen towards the electrode. And over this, there is a transport limiting membrane that facilitates oxygen transfer versus other species. So we can show here the cathodic response. We were able to design and build a system and again, file patents around it for indwelling these devices in animals and potentially in humans. But here comes the rich data part now. So one of the consequences of an indwelling device such as this is that you can generate lactate, glucose, potassium, oxygen, and pH data, and that data is temporal. That is, when the patient is first encountered, and this device, for example, placed in the muscle, we're beginning to monitor this, but the patient may be in a course from hemorrhage towards hemorrhagic shock, so that these parameters are in fact changing. And how does that then change the status of the patient? Well, we came up with a scoring system that would allow us to stratify patients into low, guarded, elevated, high, or severe. That has to do with clinical intervention. We call that stratification a HIS score, a hemorrhage intensive severity survivability score. So HIS has the potential for telling us in a fused way how all of these indicators are behaving and how they are moving. So we knew a priori going in that these parameters are within certain bounds. But I'll tell you, colleagues, there is no clear quantitative relationship between lactate and glucose. We know of the Cori cycle, and up until just 2020, here is this profound paper from Liu et al. That's really delving into the Cori cycle. We cannot write a series of differential equations based on the enzyme on-off rates, for example, that would allow us to calculate what the glucose level would be if a lactate level is such. But we know that there is a relationship, and we can exploit that relationship in building systems. Same is true for potassium and pH. We know of this empirical relationship established back in 1956 between the potassium concentration and pH in tissue. And so we could use those to build what we call sensible, fictitious, rationalized patient data. Why do we have to do this? We could not get access to clinical data that brings together glucose, lactate, pH, potassium, and oxygen. We could not. The challenge was access to the hospital system, access to the profile of the data, these specific analytes, which we believe to be instructed based on our best understanding of the physiology of hemorrhagic trauma. So we build a fictitious data set, but we rationalize this digital patient data by, for example, using these relationships that we know. So for example, here's fictitious patient number 1. We had a glucose level of 70, a lactate level of 2.7 millimolar, a pH of 7.42, that is some near normal, but somewhat alkaline. Potassium level of 5.1 and an oxygen level of 70. Well, we could do that for a set of data corresponding to 75 known different cases and 25 undefined cases. Why 75 and 25? So that we can begin to now train these data sets. So from our fictitious data, we would invite clinical experts to score the data. That is, we're going to fuse the clinical expert classification of those patients, and then we're going to use support vector machines, etc., to begin to analyze the data and stratify the data into those parameters that we talked about: low, guarded, elevated, high, and severe. So here is, for example, our first attempt with fictitious patient number 1. Fictitious patient number 1, you've just seen, 78, 2.7, 7.4, 25.1, 78. And they're being scored by 5 clinicians. Now, we're going to ask clinicians to take their time, their precious time, and go through 75 unique cases and score. The physicians, 4 of them scored this particular patient 1, guarded, and 1 scored this patient low. Immediately you see that amongst clinicians, and these are emergency room clinicians who work in trauma, there is already evidence with patient 1 of some variation in clinician status. Well, we do that for a number of patients, 75 patients, fully defined and 25 undefined. So the question is, which classification algorithm is best suited to challenge this fusion score? And what is the minimum data set that would be needed to achieve, let's say, 99% or 99.9% in the patient stratification? We use our support vector machine, and we use decision tree ensemble bag. And we saw that with both support vector machines and ensemble bag decision tree, we see that each of these classes, 0, 1, 2, 3, and 4, the 4 classes for stratification, we see that amongst the clinicians, we have different scoring. We see that if you look just by inspection, you would think that towards the middle, where the patient or sub is unsure whether they're on the safe side or on the severe side, we see the most variation in scoring. 5 scored by clinician 1, 18 clinician 2, 7 clinician 3, 15 and 13. But what we found was that we had very good accuracy with the support vector machine for clinician 2 and clinicians 4 and 5. Clinicians 1 and 3, not so much. Ensemble bag gave us, again, good scoring accuracy with clinician 2. This is beginning to reflect a little bit of the experience of these clinicians. And I can tell you more, but that's still the subject of another paper. When we look at the class, which is most confounding and presents the most confusion in our confusion matrices, we found that it was class 4 for these 2 clinicians and class 2 for these 3 clinicians. So the class designation, the stratification that is most variable was so for clinicians 1 and 2, that was 4, and for clinicians 3, 4, and 5, that was class 2. Well, we then introduced artificial neural nets with Bayesian regularization. This is classic, 5 inputs, 6 hidden layers, and the outputs. We trained these on different data sets, and we found that we needed to train our artificial neural net on what we called sorted data. That is, we could not use all of the data. The data need to reflect all of the conditions that were to be covered. All of the scoring conditions that were to be covered needed to be represented in the neural net training. And that's not surprising. When we did, however, we found that our average accuracy for the artificial neural net was about 92%, plus or minus 0.0%. With very little significant difference between the 3. We then went to possibility rule-based function approximation, which is a technique for classification based on possibility analysis. We did a 5-fold validation to come up with a mean accuracy in the test group of 0.92. So again, we find that support vector machine, ensemble bag, decision tree, artificial neural net, or possibility functional approximation all gave reasonable accuracy with very little statistical significant difference amongst them. What was interesting was to look at clinician classification. So here we're looking at the 5 clinicians in different colors, and we're looking at their majority vote in black. When we look at the type of algorithm used, not much difference. But when we look at the misclassification rate, the misclassification rate gave more insight into misclassification based on variability. So to summarize, we went further and we were able to classify our fictitious data set into low guarded, elevated, high, and severe with very good accuracy based on confusion matrices and off-axis misclassification. And we went further and analyzed the data, asking what would it take to achieve 99% accuracy? And that turned out to be a data set of 147 with 7 clinicians bringing their expert assessment. And to achieve 99.9% accuracy, we would need a data set of 154 with 9 clinicians bringing their expert observations to the data set. That we thought was quite interesting. You remember, of course, that there's very popular websites like second opinion websites. Second opinion, you're often asked to get a second opinion if you're meeting a clinician. This is not a knock on clinicians. This is just the reality of our humanity. The reality of our humanity. And if you wanted to get 99.9% accuracy in the stratification using the data set and the stuff that you would need to consult with 9 clinicians and be part of our data set of 154. So let me acknowledge some of my key collaborators in this work. All of our implantation studies were done with our colleague at the Triple Army Medical Center in Honolulu, where the implant studies were done. My colleague who began with me at Virginia Commonwealth University and is now at University of Michigan, has been involved in many of the clinical and his scoring work. And of course, my colleague Ed Church, who was at MUSC before now being at the Research Institute in Asheville, was a close ally in our discussion of first responder use of these kinds of devices. So let me summarize what we've learned. A data set of 75 and 5 clinical experts seem to be adequate for at least training and validation of our various types of machine learning algorithms. The cross-validation accuracies show no statistically significant difference whether using a support vector machine all the way through possibility analysis. But to get to higher accuracy, you need larger data sets. Now, this is not big data. This is highly precise data. And in fact, engineered data. HIS, in fact, seems to be a very useful, clinically relevant indicator that brings together these quite disparate data sources, glucose, lactate, potassium, oxygen, etc., into a fusion, through a fusion algorithm that gives you a classification. And so we recently published this in a electronic textbook.
I'm sorry, I have to interrupt you. We have 20 seconds before we have to end the session.
Okay, good.
Thank you so much for the nice talk. And if you have any questions, we don't have time for Tony to answer questions, but you can always input your question into the chat room. And I think Tony, we can get back to you. Sorry for that, because we have to end in time and then catch another session. Just thank you so much, Tony. Yeah.
Thank you so very much. And I apologize for running over.
No, it's good. Great talk. Yeah. Thank you so much.
You guys can post questions in the chat box on Kuba, and Dr. Guiseppi-Elie can answer them there. Thank you, everybody.
Thank you.